Branco, and the title is Ecological Factors Affecting Plant Community Distribution on Installation in Tagus River Natural Park. Straddling the frontiers of the neighbouring countries around Tagus River, Portugal and Spain, the International Tagus River National Park extends over an area of 36,484 hectares in the district of Castelo Branco, Portugal, near the border with Spain. The vegetation of the International Tagus River National Park is mainly typical of Mediterranean ecosystems, such as evergreen scrubland, dominated by holm and wild olive trees, with a mosaic of thermophilic formations, bush formations, and riparian vegetation. In the territory, 736 hectares, distributed by 98 botanical families, have been identified to date. The size of the 51 and 10 is detected of plants of growth. The park, the Tagus River, International Tagus River Natural Park, well known in terms of its composition, including more than 200 species of vertebrates. So far, 179 species of birds have been invented in the park area, some of them with a high conservation state, such as the black stork, the imperial eagle, that's right, the black vulture, and the Bonellis eagle, and also the Egyptian ocean. The objectives of this study: first, to implement the field characterization of the 188 plant communities, evenly distributed along the park, representing homogeneous vegetation types, with different homogeneous vegetation types. Other objective is to identify the main environmental factors that influence the distribution of plant species and communities. 3, to give useful information to stakeholders in order to manage the natural and agroforestry production areas inside the natural park. And 4, to understand the influence of the present climate conditions in order to study future trends related with the climate change provision, the impact of flora and vegetation for this area. Methods used. First, in the field survey, the vegetation data were collected at each site. 188 sites will be shown in the next figure. All vascular plant species were surveyed and species identification was carried out. The information recorded at each site including plant species, abundance, coverage of each species, and physical attributes of the site. Growth aspects on property, but of course, those.
Do we want to do this whole one? Because it's like, it's so difficult to understand. What do you think?
In the mapping, the cartography that we have for the area. We have
how long it is more?
It's a 20-minute video. It's 20 minutes long. It's just his microphone. His microphone was not configured correctly. You know, it's not. I tell you, in 2021, we'll all have it down to do microphones and sharing and everything. Well, we do have, oh, Dr. Garancha
Yes.
Goljubovic is here. So was that the video we were just showing? I'm sorry. We also have the poster now from the one on interferometers. I can show that if we want to do that. Or do you want to talk a little bit about your presentation on the vegetation? Is that person here?
I'm sorry.
Oh, okay. So how should we proceed? Do you think, I think maybe for the video, people could watch it on their own, and because the audio is not very useful, they might just want to look at the slides in that video. It is shared.
You want me to present
oh, you're here. Yeah, yeah.
Instead
right,
of video. Because it was a video that you were.
Oh, did we ask for a video? I'm sorry. I meant to, what I was saying was, yeah, your audio sounds much better now. Do you want to talk about
okay. I could present, make the presentation. It's a problem of course. Wait
Thank you.
No
sure, yeah, go ahead, I guess. Yes.
Okay, wait a minute, please.
I'm sorry, I just got a little confused, so now I understand.
Okay. No problem.
Are you working on sharing your screen, or?
Yes, I'm open the PowerPoint.
Okay, excellent.
Okay. Okay, now I am here.
Yes, now we have it.
That I begin the presentation from the beginning, or?
Yeah, I suppose.
Yes. Okay.
But we're down to a small crowd, so we only got 9 people left, not counting myself and Ismail.
Okay, I will try to go fast. Okay. Yes.
Yes.
The study was about the relationship between the ecological factors and the plant communities distribution in the International Tagus River Natural Park. That's a park that is located near the border with Spain. The Tagus River, it's a river that it's a border between Portugal and Spain. We have protection measures in the Portuguese side, and the Spanish has also measures in their side. You are listening? You are listening well?
Yeah.
Okay, that's good. Okay, and this is some information about the area. The area of the natural park is 26,484 hectares in the district of Castelo Branco. You can see in the map. The vegetation is typical Mediterranean type vegetation with a system that in Spain they call dehesa, and in Portugal we call montado. That's a low density forest with oak trees, perennial oak trees like holm oak, and sometimes also cork oak, and olive trees, wild olive trees, with a mosaic of thermophilic formations, bush formations, different types of vegetation. And in this territory, 726 taxa distributed by 98 botanical families have been identified till now. It's also well known, and it's protected essentially because of some birds of prey and buitres that exist in the park with a high conservation status. One is the imperial eagle, the black vulture, the Bonellis eagle, and the Egyptian vulture. Some of the species that have high conservation status. What are the objectives of this study? To implement a field characterization of 188 plant communities, evenly distributing along the park, representing different homogeneous vegetation types. To identify the main environmental factors that influence the distribution of plant species and communities, environmental factors. To give useful information to stakeholders in order to manage the natural and agroforestal production areas inside the natural park. And also to understand the influence of the present climate conditions in order to study the future trends, modeling different scenarios of climate change related with the provisions and its impact on vegetation. Field survey. The vegetation data were collected in the 188 sites. All vascular plant species were surveyed and species identification was carried out. The information recorded at each site included plant species, abundance and coverage of each species, and physical attributes of the site that were complemented with the information in the maps, the cartography. We use GIS, geostatistical tools of GIS, to interpolate the species distribution and the community species richness. Richness is a measure of diversity, as you know. Cluster analysis was also made to complement not to complement it, it was ordination analysis was also made to complement the cluster analysis, was used in order to aggregate the main vegetation communities based in its composition. The ordination analysis, it was used the canonical correspondence analysis, CCA, to relate the environmental factors with the distribution of different species. Here you have the location of the sites of the floristic inventories. As you see, it was more or less distributed by the protected area. Results for the field survey, 309 vascular plant species were identified in the totals of sites. In terms of GIS modeling, wooden species distribution and species richness maps in the International Tagus River Natural Park were produced. I will show some of them. The cluster analysis. With the cluster analysis, we identified 5 distinctive plant communities based on the species composition. And in terms of ordination analysis, CCA shows that the most important factors affecting the frequency of plant species in the park were the slope, elevation, and soil type, topographic and soil factors, essentially. Here you see the distribution of the species richness, the diversity of plants. Here is the distribution of one species, the main tree, the Quercus rotundifolia, the holm oak distribution. Here, other plants, mastic, Pistacia lentiscus. Other species from the same genus, Pistacia terebinth, distribution completely different. Here, the 2 subspecies of the same species, Lavandula stoicus, the Spanish lavender. They have this subspecies Luisieri as a distribution completely different from this subspecies Sampayana. Here we have the results of the cluster analysis and the 5 communities that were identified. Here you have an explanation of the composition of each community. The community 1, it's composed by different riparian vegetation types, dominated by bushweed, Flugic tinctoria, belonging to this sociological community that I will not say the name because most of you are not from this area, and also dominated by sage-leaved willow, a type of willow, a species of willow. The community 2, it's natural grassland communities, essentially, different types of herbaceous communities. The community 3, it's a low density holm oak forest with some species in the soil, small bushes of different species. The community 4, it's constituted with scrubs. It's frequently associated to a low density holm oak trees dominated by gum rockrose, Cistulosa ladanifer. And the community 5, it's tall scrubs dominated by species like Ratama, broom, Ligus phariocarpa, and the Portuguese broom, Cytisus striatus. The last analysis, it was the CCA. It's a exploratory multivariate statistical approach. And the objective is to identify the main environmental factors that affect the species distribution. We used as environmental factors the bioclimatic units that were defined by the thermicity index and the ambrothermic index, two bioclimatic indices. Topography, we use elevation or altitude, slope, and aspect. And also a classification of soil types, considering the structure, the texture, the pH, the organic matter, active calcium, and carbon nitrate ratio, all these different properties of the soil. Here you have the maps with the distribution of the bioclimatic units, the digital elevation model that was used to also to not only for altitude, but also for slope and aspects, and the soil types. Here you have the CCA biplot that has a significant correlation between the 5 variables that were used that appear in ordination axis at red. And the most important, as I already told, the most important factors affecting the frequency of plant species in the park area were slope, elevation, and soil type. And the total of the variance explained with the first 2 axes is very good, 71.29%. Final remarks. This is an ongoing study. The combination of multivariate analysis and GIS can therefore be seen as useful tools in vegetation ecology studies. Topography and soil are the most influential factors that explain the species distribution in the park. The study results will be helpful to stakeholders in order to manage natural and agroforestry production areas inside the natural park, and to make, for instance, vegetation after fires, wildfires, would be important to know the species that they can use. And future studies will be to see the impact of the climate change on flora and vegetation. Thank you very much.